Imagine a world where creatures long thought lost to history walk the earth once again. From legendary beasts to ancient wonders, scientists are rewriting the rules of extinction. Stay tuned because today we're diving into 10 extinct animals making an unbelievable comeback in 2025. Let's begin.
Woolly rhinoceros. The woolly rhinoceros, an Ice Age giant with a thick fur coat and massive horns, roamed Eurasia's tundra until it went extinct 14,000 years ago. In 2021, scientists at the University of Stockholm extracted DNA from a 20,000-year-old rhino carcass preserved in Siberian permafrost near the Copacabana-Karga River. Using CRISPR, they added genes of the critically endangered Sumatran rhino, its closest living relative, to add cold-resistant traits like dense fur and fat layers. A 2023 in Nature Ecology and Evolution identified the TRPV3 gene, which regulates heat sensitivity, as key to surviving Arctic winters. Colossal Biosciences aims to create hybrid embryos by 2028, with plans to release herds in Siberia's Pleistocene Park. These rhinos would trample snow, exposing soil to freeze and lock in methane, a greenhouse gas 80 times stronger than CO2. A 2022 UN report warned that thawing permafrost could release 1.7 billion tons of carbon by 2100. Challenges include protecting hybrids from poaching, but the project aligns with Russia's 2030 Arctic conservation strategy.
Tvrti dodo. The dodo, a flightless bird from Mauritius, was wiped out by 1690 due to sailors and invasive rats. In 2022, scientists at UC Santa Cruz, led by evolutionary biologist Beth Shapiro, fully sequenced its genome using a 400-year-old skull from the Copenhagen Natural History Museum. Partnering with Colossal Biosciences, they now edit genes of the Nicobar pigeon, the dodo's closest relative, to recreate features like a robust beak and reduced wings. CRISPR targets genes like BMP2, which controls wing growth, to mimic flightlessness. The Mauritian Wildlife Foundation plans to reintroduce dodos to predator-free islands like Île aux Aigrettes by 2035. A 2019 study in Science showed the endangered Tambalacoke tree's seeds only germinate after being scarified by dodo-like digestion. The project uses funding from the IUCN's Endangered Species Recovery Fund and collaborates with local communities to remove invasive species.
Dire wolf. The dire wolf, an Ice Age predator featured in Game of Thrones, vanished 10,000 years ago. In 2025, Colossal Biosciences, led by geneticist George Church, merged DNA from dire wolf fossils found in California's La Brea tar pits, a site holding over 4,000 dire wolf skeletons, with gray wolf cells. Using CRISPR, scientists built genetic caps to create hybrid pups named Romulus, Remus, and Khaleesi, a nod to Game of Thrones. These wolves have thicker fur, stronger jaws, and a stockier build, traits pulled from ancient DNA. The goal is to reintroduce them to North American forests, where they could control deer overpopulation and restore ecosystems. The project builds on a 2021 study published in Nature, where CRISPR was used to edit wolf DNA for the first time. Colossal's work also references Ice Age predator roles documented in the La Brea Tar Pits Museum, showing dire wolves hunted megafauna like bison. If successful, this could reduce deer-car collision and overgrazing, issues costing the U.S. $1 billion annually.
Steller's sea cow. Steller's sea cow, a 30-foot marine mammal, was hunted to extinction by 1768, just 27 years after its discovery near Alaska. In 2021, the University of Alaska sequenced its DNA using bones from Bering Island, where the last herd lived. Scientists now edit genes of the dugong, its closest relative, using CRISPR to replicate traits like thick blubber and kelp-digesting enzymes. A 2023 marine biology study identified the LCT gene, critical for processing kelp sugars. The plan is to reintroduce sea cows to Alaska's Aleutian Islands by 2035, restoring their role in kelp forest ecosystems. These forests absorb 20 times more CO2 than terrestrial forests, as per a 2022 UNESCO report. Challenges include protecting sea cows from ship strikes, but drone-monitored kelp corridors are being tested. If successful, they could revive a species lost to human greed and inspire marine conservation efforts worldwide.
Saber-toothed cat. The saber-toothed cat, Similodon, an ancient ice predator, went extinct 10,000 years ago. In 2023, Colossal Bioscience has launched a project to recreate its traits using CRISPR-edited DNA from fossils found in California's La Brea tar pits. Key genes like BMP3 linked to tooth growth are merged into African lion cells to develop embryos with saber-like canines. A UCLA study published in Science Advances 2022 confirms Similodon's DNA shares 95% similarity with modern lions. However, ethical debates persist. Reintroducing an apex predator risks conflicts with humans and livestock. The project focuses on lab research with no plans for wild release. Science aims to study Similodon's hunting behaviors using virtual models based on fossil biomechanics. If successful, it could reveal how Ice Age predators shaped ecosystems, as noted in a 2021 La Brea Tar Pits Museum report. The work also aids big cat conservation, like understanding genetic diversity in endangered Siberian tigers. Quagga. The quagga, a zebra subspecies with stripes only on its front half, went extinct in 1883 after being hunted for its hide. In 1987, South African biologist Reinhold Rauh analyzed DNA from the quagga skin at the Natural History Museum of Zimbabwe, proving it was a subspecies of the plain zebra. This led to the Quagga Project, which selectively breeds zebras with quagga-like traits. By 2005, the project produced Henry, a zebra with 90% quagga DNA, confirmed by a University of Cape Town study. These quagga-like zebras now graze in South Africa's Cairo National Park, where they control invasive grasses, as noted in a 2020 report by South African National Parks. The project aims to fully restore the quagga by 2040, using DNA from 23 museum specimens. In 2022, researchers used CRISPR to edit zebra embryos, speeding up the process. The quagga return could revive its role in grassland ecosystems, reducing wildlife risk by limiting dry overgrowth. Moa. The moa, a 12-foot-tall flightless bird, was hunted to extinction by New Zealand's Maori people around 1445 AD. In 2018, Harvard scientists sequenced its genome using DNA from eggshells found in caves. Published in Proceedings of the National Academy of Sciences, the University of Otago's de-extinction project edits emu embryos, the moa's closest relative, to activate genes like COL2A1, which govern leg growth. The goal is to reintroduce moa to Fiordland National Park by 2040, where they disperse seeds of trees like the katiak. As described in a 2020 ecological study, critics argue funds should protect living birds like the kiwi, but proponents cite the moa's role in Maori culture and its potential to boost ecotourism, which earned New Zealand $5 billion in 2022. The project uses lessons from New Zealand's successful Kapau parrot recovery program, combining CRISPR with habitat restoration. If achieved, it would mark the largest bird revival in history. Tasmanian tiger. The thylacine, a striped marsupial predator, was declared extinct in 1936 after decades of hunting. By 2017, scientists at the University of Melbourne sequenced its genome using DNA from a pup preserved in alcohol since 1909, stored at the museum's Victoria. Partnering with Colossal Biosciences, they edit cells of the fat-tailed dunnart, the thylacine's closest living relative, using CRISPR to activate dormant genes for jaw structure and striped fur. The first hybrid embryos could be ready by 2025, with plans to reintroduce thylacines to Tasmania by 2030 under Australia's Wildlife Restoration Initiative. If successful, thylacines could control invasive species like rabbits, which cost Australian agriculture $200 million yearly. In 2023, the team grew thylacine muscle cells in a lab, detailed in Nature Communications. The project aligns with Tasmania's 2020 biodiversity strategy, aiming to restore native predators. Thylacines once played a key role in balancing ecosystems, as noted in 19th century journals by naturalist George Harris. Aurochs. The aurochs, a massive wild cattle species depicted in France's Loch cave paintings, went extinct in 1627. The European Tauros Programme, launched in 2008, used primitive cattle breeds like Spanish Sagese and Italian Marimana to backbreed aurochs like animals. A 2015 study in Nature compared their DNA to a 6,000-year-old auroch bone found in Germany, confirming 80% genetic overlap. These tauros now roam rewilding areas in Portugal's Cow Valley and the Netherlands' Mahorst Nature Reserve. Their grazing prevents forest overgrowth, reducing wildlife fire risk by 40%, as reported in the Dutch government's 2021 rewilding plan. The project collaborates with the University of Wageningen to track ecological impacts using GPS collars. By 2035, the goal is herds that mimic auroch behaviors, like defending against wolves, as described in Julius Caesar's Gallic Wars. Restoring aurochs could revive Europe's ancient grasslands, critical for endangered species like the European bison. Woolly mammoth. Woolly mammoths vanished 4,000 years ago, but Colossal Biosciences aims to revive them using CRISPR. Scientists edit Asian elephant DNA with genes from a 28,000-year-old mammoth named Yucca, discovered in Siberia in 2010. Key traits like shaggy hair, small ears, and fat layers are added for Arctic survival. Colossal previously engineered a woolly mouse with mammoth-like traits, signifying the feasibility of their approach. The plan is to release herds into the Pleistocene Park, a 20,000-acre reserve in Russia founded by scientist Sergei Zimov. Here mammoths would trample snow, exposing soil to freeze and trap methane, a greenhouse gas 25 times stronger than CO2. In 2022, Harvard researchers led by George Church grew mammoth-like stem cells in the lab, as published in Science Advances. This proved mammoth genes could function in living cells. The project also uses permafrost-preserved DNA from the Northeast Science Station in Siberia. If successful, these hybrids could slow Arctic permafrost melt, which holds 1.5 trillion tons of carbon. This marks the first attempt to use an extinct species to tackle climate challenges.
Which animal would you love to see roaming our world again? Tell us in the comments below. If you enjoyed this wild journey through time, make sure to hit that like button, subscribe, and turn on notifications, because the future of wildlife is just getting started. Stay curious, stay amazed, and we'll see you in the next adventure.